Hello, everyone. So good morning for some, good afternoon for others, good evening for others as well. We'll just, people are just trickling in, but also we are live streaming this, so you have the opportunity to, we will get started, but if you're late joining, you can watch what you've missed at the end, as it will also be available on demand. So thank you to everyone for joining today. We have two really exciting speakers who will be talking about an initiative called Target 2035 within the context of small molecule drug discovery. So before I get into introducing the speakers, as I mentioned, it is being recorded. So we're live streaming on LinkedIn right now, and will be available on YouTube on demand afterward. So feel free to ask questions throughout the session. Use the chat box, and I will make sure that your questions are answered by the speakers. As well, there will be some polls taken throughout the episode. Oh, Vijay, could you please jump to the next slide? Thank you. So this is just introducing our molecule to medicine, or as we call it, M2M series roadmap. So we are currently on episode 4 of this series. We actually will be extending it beyond episode 8. So we'll be carrying this out throughout all of next year. So stay tuned for the remaining episode titles and themes. This is due to the interest and that we've had throughout, and lots of speakers have come forth and wanted to talk about new things. So we're going to continue along, and hope you're enjoying what you're watching so far. Thanks, Vijay. If you could just go to the next slide. So I'll introduce our very own Vijay Shahani. So he's the director of applied science at Cyclica. And he helps inform on the development of the tools and their applications in drug discovery research. So he leads a team of scientists at Cyclica, the applied science team, and they apply Cyclica's technology to a diverse range of drug discovery programs. Vijay received his PhD from the University of Toronto using computational methods to design and synthesize small molecule inhibitors for proto-oncoprotein. And following his doctoral studies, he went on to receive his master's in biomedical communications also from University of Toronto. And so he explored the use of multiple representation of complex concepts to facilitate learning. So thanks so much, Vijay, and I will let you introduce Matthieu himself.
Thank you very much, Jen, and very happy to introduce Dr. Matthieu Schapira, who I've had the great pleasure to work with on a few projects while at Cyclica, including an NSERC-sponsored project exploring SARS-CoV-2 pocketome for opportunities to develop a pan-coronavirus or pan-variant inhibitor or drug potentially down the line if it all works out, and which is, I guess, a very topical thing given the environment that we're in right now. And as well as some work that we did together on a quote-unquote undruggable decaf-1 protein, which we will touch on a little bit later. Just a little bit about Matthieu. He's received his master's in chemistry and PhD in biochemistry in France. He then went on to complete a couple postdoctoral fellowships in protein crystallography and computational chemistry at the New York University Medical Center. After leading structure-based drug design at biotechs in both San Diego and France, he joined the SGC as a PI in 2007. He is also an associate professor in the Department of Pharmacology and Toxicology at the University of Toronto. Very pleased that Matthieu will be here to discuss the importance of small molecule probe development and how that impacts drug discovery, as well as some of the key initiatives he's involved with at the SGC, including Target 2035 and also CASP, which I'm excited to hear a little bit more about. So with that, I think I'll turn it over to Matthieu, who's prepared some slides for us, which we'll work through together. But Matthieu, the floor is yours.
Thank you, Vijay. It's great to be here. And thanks for the introduction. So I would like for us to discuss Target 2035, which is a large-scale project, comparable in size with the sequencing of the human genome 20 years ago. And well, the vision is to make a chemical probe or a chemical tool for each druggable protein encoded in the human genome. So why would we want to do this? So to understand this, let's take a step back and look at the efficiency of biomedical research. So as I said, the human genome was sequenced, well, we can stay on this one. Yeah, the human genome was sequenced 20 years ago, over 20 years ago. And every year, about $250 billion are invested in biomedical research from the public and the private sector. And still, we don't understand the molecular mechanism for many diseases, including common diseases such as Parkinson's disease. And when we do understand this molecular mechanism, the medicine that we make are more often than not affordable for most people in the world. So there is a problem in efficiency and in cost effectiveness, obviously. If we go to the next slide, we can see what we believe is one of the reasons for this lack of cost effectiveness in making affordable medicine, which is redundancy. So if you plot, so on this plot, you can see on the x-axis all genes from the human genome, about 20,000 of them. And on the y-axis are the number of publications for each of these genes. So you can see that we are all working on the same 10% of the human genome, and very much ignoring the remaining 90%. And actually, 35% of proteins encoded in the human genome are absolutely not characterized at all. On the other hand, if you look at unbiased metrics, such as genome-wide association studies at the top or the OMIM database, you'll see that many of these underexplored proteins seem to be disease-associated, and very probably some of these genes and proteins are valid targets for unmet medical needs. It's just we are not yet aware of it, and so we are all working on these 10% of the human genome and missing opportunities. And this has a cascading effect all the way to clinical trials. So as you probably all know, the number one reason today why a drug fails in phase 3 clinical trial is not for toxicity, it's for the lack of efficacy. So the compound, the drug is doing exactly what it was designed to do molecularly. So it is inhibiting the target, the protein target that was selected. The problem is that inhibiting this protein target does not have the beneficial medical outcome that was expected. So the problem was that the target that was selected was not the right one. And probably if we were looking at characterizing better these underexplored protein, we would find better targets leading to better drugs. So if we go to the next slide, probably one of the main reasons for this, for the fact that we are ignoring a big portion of the human genome is for the lack of tools. If you don't have a specific antibody for a protein, you will not be able to do a simple Western blot. Another type of tools that turned out to be very much enabling are chemical probes. So if we go to the next slide, chemical probes are compounds that can potently, selectively engage a protein in cells. And in doing so, it can be used to link a gene to a phenotype. And additionally, once you have identified a chemical probe, so it's a drug-like molecule that does not have necessarily all the properties that you want that the drug needs to be. That doesn't have. We don't focus on the ADMET, for instance, and in many cases these chemical probes are used for assays in cells, not in vivo. But they do provide starting points for a ligand-based design to find other compounds that can support drug discovery. So let's go on the next slide.
Matthieu, if I may, just I think one of the things that you pointed out here, which I think is really important and why chemical probes might serve as a great transition point for drug discovery, is the fact that you're engaging with proteins and some of the other ways that people explore the consequence of a protein is by doing knockout or knockdown studies. And unfortunately, that's upstream. That's working with the translation of mRNA to proteins. But when you think about drugs, for the most part, they work on proteins. And so chemical probes probably give you a little bit of, and maybe you could correct me if I'm wrong, but it gives you a little bit of a better sense of what you would actually expect if you were engaging with this target with a small molecule drug, per se.
Yeah, absolutely. Yes. If you knock out or knock down a protein and a gene, and then you remove entirely the protein, and so any structural or effect of the protein or any function of the protein that is not related to the specific site that may be targeted by a drug will also be erased, and so with potential phenotypic outcomes that are not related to what you would see that elicited by a drug. So in this regard, a chemical probe is much more reflecting much more accurately what you should expect to see from an optimized drug.
Great. Okay. Thanks, Matthieu.
So because so at the Structural Genomics Consortium, because we are quite convinced that these chemical probes are very much enabling tool, in the past 10 years, we have conducted a pilot study, if you will, to make chemical probes focusing on epigenetic target classes. So these are proteins that are involved in chromatin-mediated signaling that regulate transcriptional events. And so we have, since 2010 we have partnered with 10 pharmaceutical companies, medicinal chemists from 10 pharmaceutical companies, to make what is kind of old slides, old numbers, way over 60 chemical probes that are open science, so they are available as commercial vendors with no restriction on use. And because these chemical probes are high quality and open science, they are very popular. So over 5,000 samples are distributed each year to enable drug discovery in laboratories, both in the public and the private sectors. So all these research groups are using our chemical probes to ask their own question using their own assays, their own cellular models, and come up with unexpected findings that they publish. So that's over 3,000 publications since 2011, and some of these publications reveal novel gene-to-disease links that in turn led to over 30 clinical trials by 8 companies. So very significant impact on this pilot project. So so on the next slide. Yeah, so the next
Sorry,
step.
Matthieu, just one quick note before we move on. I just wanted to quickly, again, significant impact, like you said. It's, congratulations, these numbers are quite encouraging, obviously very exciting, especially that 30 clinical trials launched. So it's definitely shows the impact. And I think the other key thing here is SGC works with pharma. And so it kind of points to the significance that this work has for the entire industry. I'm not saying that everything that pharma does necessarily means it's good, but I think it does point to the fact that this is a significant aspect of the drug discovery process, and support is needed. And I think that's, what you're going to present next really speaks to kind of the vision and thinking ahead to what kind of impact the next few slides will have. So I just wanted to chime in with that.
Yeah, absolutely. It's kind of, at first, it's kind of counterintuitive to think that all these pharma companies would be working on projects that are open science with zero, any results from the SGC. None of the results from the SGC is patent protected. It's really putting all this knowledge, including the structure of the compound, in the public domain. And this really speaks to the fact that that the pharmaceutical companies are agreeing that the knowledge that these chemical probes will generate by being used in laboratories around the world can be very beneficial to them, because then it can reveal novel protein targets, novel therapeutic opportunities, novel mechanism to go after unmet medical needs, and yeah, so this is quite compelling, I think. Okay, so the next step, since there was a strong impact here, the next step for us is to expand from this from this large, but it was still a pilot study focused on epigenetic target classes, to expand to the entire human genome. And so this is what we call Target 2035. So the goal is to make a chemical probe or a chemical tool for all human druggable proteins within, well, 13 years from today, I guess. Of course, there is no way we're going to do this on our own at the Structural Genomics Consortium. And so the first step is to initiate an international federation of biomedical scientists from the public and the private sector who are ready to contribute to this effort. And even together, we will not be able to achieve this goal with today's technology. So the first phase of this initiative is really to develop and identify new technologies to more efficiently identify hits and optimize them. And also establish a roadmap to coordinate this global effort. And once this is in place, then the second phase will be more of a production phase, if you will, where we'll have to churn out all these clinical probes together. So if you are interested in knowing more about this initiative, well, first you can read the white paper that was published in 2019. You can see at the bottom of this slide. And the follow-up paper that just came out a couple of weeks ago in RSA MedChem. And you can also definitely you go to the website, target2035.net, where you can see how you can join the effort. And also you can attend monthly webinars that are each focused on the different technological aspect or different protein family. I believe on the next slide, a few are listed just as examples. These are available after, I mean, they are both, you can attend them live, but also attend them, view them on YouTube whenever you want. So for instance, you can see that some of the past webinars were focused on computational AI for drug discovery, on covalent inhibitors, chemoproteomics, or drugging pseudokinases, for instance. All right, so maybe we can go to the next slide.
Yeah, maybe before we do, Matthieu, just to pick your brain a little bit, I see the word druggable being used quite a bit, a druggable protein, and I think everyone might have a slightly different notion of what a druggable protein is, and I'm not sure how that's changed with the advent of targeted protein degradation. But I wouldn't mind hearing from you. How do you gauge druggability, or if a protein is druggable or not?
Yeah, so it's a moving target. It's a moving target. There's no valid or definitive definition of what is druggable. One way of looking at it is computational, looking at the geometry of a binding site. It's looking whether, I mean, typically a pocket is, the druggability of a binding site is based on its enclosure, its volume, its hydrophobicity. So there are some tools out there that are used regularly to predict the druggability of the binding site, and maybe one of the most common tools is sitemap from Schrödinger. There are also ways to interrogate this experimentally. For instance, if you do a fragment screen on a pocket, probably you'll see a site where you have multiple fragments that are clustered in your pocket. If you do this crystallographically, and this can indicate also a chemically trackable site. As you mentioned, Vijay, what was believed to be undruggable yesterday is believed to be druggable today with PROTACs, which are compounds that can stabilize the interaction between two proteins, and that can exploit sites that are functionally neutral on a target protein. By using PROTACs, you can have a protein with a functional site that is where the catalytic site and another domain. Maybe your catalytic site is undruggable, but maybe this additional domain is druggable, in which case you can make a PROTAC that will bind to this domain, recruit an E3 ligase that will ubiquitinate the protein, leading to its degradation. So yeah, again, there is no definitive way of evaluating the druggability of a pocket. So it's a tough one. You can also go by protein families. For instance, some protein families are well known to be druggable. So this is one way of looking at it. Kinases are druggable, and other families may not be as well.
Right, and then that also leads to the same problem that you talked to earlier, where research seems to be focused on areas where there's been success before, right? And so kinases historically lots of success there. GPCR is potentially getting more and more popular, but it leads to a bias where all your data that you're generating, and this is maybe to putting on the AI or ML hat, a lot of your data that you're generating is for the same target classes and for the same families, and so your applicability to those, other potentially therapeutic targets that are understudied, is somewhat diminished. So the aspiration of Target 2035, where you're going to be exploring all druggable proteins, which would be including obviously the ones that are known to be drugged, but also those that are less studied, I think it have a major impact on the performance of these ML engines. And maybe one other question about this, and how does one, obviously the aspiration is all druggable proteins, but is there a strategy to the types of proteins that you're going to focus on first, or is it more emergent based on where the research is and what's available?
Yes, so different. So yes, already quite a few large consortia are starting to contribute to Target 2035, and each have their own focus, which is often target-based focus. When you start learning how to make kinase inhibitors, then you're getting better at making kinase inhibitors, and then you'll be more efficient at making inhibitors for new kinases. So same thing with in the case of epigenetic target classes, where the Structural Genomics Consortium worked a lot. For instance, in Toronto, we were focusing on and still a little bit are focusing on protein methyltransferases. And at one point, you start understanding more and more the structural chemistry of the binding site that is targeted by these protein methyltransferases, and then you can get a little bit better at identifying hits or finding strategies to optimize them. Knowing the type of libraries you might want to screen, etc. So, for instance, the consortium called Resolute in Europe is focusing on solid carriers. The IDG, the Illuminate Druggable Genome, in the US is focusing on kinases and on GPCRs. Or UB Open, another large consortium in Europe, is focusing on solid carriers and E3 ligase. And here at the Structural Genomics Consortium in Toronto, we're focusing on WD repeat containing proteins. Maybe I can come back to this a little bit later, and on E3 ligases as well. So it's mostly a target-based approach to try to be more efficient about it. But at the same time, try to I mean, it's often easier to find collaborators and to raise funds to work on proteins that have some type of seem to have some based on genetic information, some disease association.
So in those cases, a bit opportunistic. Yes. Sorry, Jen, I see you.
If I may jump in, we have a question from the audience. So
Sure.
the question is, what should be the strategy to target proteins having similar druggable sites, but different post-translational modifications belonging to the same protein family?
Okay, different post-translational modifications.
That's a great question.
Yes.
It's a stumper.
Typically, I mean, I did not so far come across a protein that was post-translationally modified at the drug binding site. Can imagine it could happen. If this is the question. Yeah, it's something that I haven't encountered personally. Interesting question. Yeah.
Yeah, very interesting, and it's maybe something that is, I think, when you're doing a drug discovery program, and especially thinking about things protein-wise, you may overlook things like post-translational modifications to some extent, given how they are often key for mediating interactions with other proteins or serves other purposes beyond maybe just the catalytic function of a protein. And so it's, I guess one thing you could do with the structural analysis is if you could co-crystallize the structures themselves, or crystallize the structures themselves, see if there's any long-range effects that might make microscopic differences in the binding sites which you could exploit. But it's a very difficult question, and I think it's very case specific, but definitely a stumper. So I'm going to think on this a little bit more, and then maybe we could circle back to it towards the end of the presentation.
Great, thank you.
Great, Matthew, maybe we'll move forward with the next slide.
Yeah, right. So so speaking of new technologies that could accelerate hit identification and hit optimization, that could, which is the first phase of Target 2035. I think it's pretty obvious that given today's, given the continuous leap in computational power, the ever-expanding size of the commercial available chemistry space, and the emerging potential of deep learning in the computational drug design, AI is very high on our radar of potential of technologies that may significantly accelerate towards the goal of set by Target 2035. So we at the Structural Genomics Consortium, we in the last year and a half, we have tried to be systematic, come up with a systematic way of asking the question: can AI accelerate hit discovery? And so to do so, if we go to the next slide, we partnered we collaborated with two biotechs, so Cyclica here in Toronto and Atomwise in San Francisco, and we focused on WG repeat containing proteins. I can tell you more about this target class if you want later. And so the way this collaboration worked, and it's still working, it's an ongoing project, is that the biotechs start from the crystal structure of a protein of interest and use their discovery engine, their machine learning, to screen millions of commercial available compounds and select 100 or 200 of these millions of compounds. And these 100 or 200 commercial compounds are then procured and tested experimentally in the group of Professor Masoud Bedari at the Structural Genomics Consortium in Toronto. And so this is the same group. This is Masoud's group, who also has worked for 10 years with all these pharma companies And he has established a very robust experimental platform where compounds are tested in orthogonal assays and quality control, testing the solubility there, and it's very thoroughly, robustly characterized. And if we go to the next slide, we can see some of the results. So for instance, on this slide, you can see this compound in the middle. It was one of about 100 compounds that were selected by Julie Owen at Cyclica. And Julie predicted that this compound would be binding to DCAF1, which is an E3 ligase. And Alice Lee in Matthieu Schapira's group at the SGC tested this experimentally and confirmed that this compound indeed was binding to DCAF1 in the SPR assay here with a KD of 70 micromolar. So it was not very potent, but it was definitely reproducible hit experimentally. And the compound was behaving well in solution. It was soluble, and because it was robust and reproducible hit, we tried to crystallize this. And so Sarah Kemani in Levon Halabalyan's group at the SGC was able to crystallize this compound bound to its target DCAF1, which is the structure that you see here, and which reveals some possible vectors of optimization. The structure also shows that the compound is sufficiently accessible from the surface of the protein that it would be used, for instance, as a chemical handle to make a PROTAC that would recruit DCAF1, which is a call for E3 ligase, for instance. On the next slide, you can see that so Julie at Cyclica then selected, based on this hit, she selected another 20 compounds, 20 derivatives that were commercially available, and one of them is a 3 micromolar compound for DCAF1. So it's quite exciting to see that just after testing experimentally, hardly more than 100 compounds were at 3 micromolar KD here. And on the next slide, you can see that we got also encouraging results from another method at atom-wise. You can see these 3 compounds identified against 3 different WG repeat-containing proteins. Again, the potency is not as good as we were initially hoping for, but in all cases, all these compounds are first in class for the protein. So there was until before that, there was no known inhibitors for any of these proteins. So that's again very encouraging, and these compounds again were quite reproducible. So at this point, we are also trying to optimize to go to a quick optimization round to gain further confidence that these hits are optimizable. And on the next slide was it was a variation on the same theme, but this time it was not a purely computational approach. It was a hybrid approach in collaboration with Zebi AI, a biotech in Boston that since then was acquired by Relay Therapeutics. And here, the protein target that was selected was produced, expressed, purified at SGC. So the physical protein sample then is used at Zebi AI to do a DEL screen, DNA encoded library screen. And then Zebi, instead of resynthesizing the DEL hits of DNA, which is the costly and long process, instead of doing this, they use their machine learning to learn from these DEL hits and from the negatives, and then apply this toward the selection of 100 to 200 commercial compounds that are then again tested experimentally in Subedari's group at the SGC. And on the next slide,
if I may ask a quick question, the method that was used for the DEL screen, is that a biophysical type of evaluation, like a SPR, or?
Yes. So the way DEL screens work is that you have a library of millions, sometimes even hundreds of millions or more compounds, and each molecule is tagged with a little DNA tag. So there's a label on each molecule, and then you have this solution of millions of molecules, each tagged with its own DNA, unique DNA tag. And then you have a protein, your protein of interest that is immobilized on a chip generally. And then the hits, the valid hits, bind to your protein, then you wash the rest. And then you use the DNA, you use a PCR to re-amplify the tags. And then so even though you have an infinitesimal amount of each of the molecules, because you use PCR to amplify the DNA tags, you can see which are your putative hits. So it's a binding assay.
Great. And then from those counts at the end of the day, that's what Zebi was able to use to predict in a different library altogether what will likely to be a binder, non-binder. And those are the ones that were
yes, exactly.
that you see.
Yeah, typically what you do once you have amplified your DNA and identify which of the compound were binding in principle, then you resynthesize the compound but without the DNA, because it's very possible that the binding is driven by the DNA tag and not by the compound, especially if the compound is about that big and the DNA is that big. So the binding event, is it this that is contributing or this contributing? You can have a lot of false positives. So but if you are smart about it and you look at the chemistry of your different hits, then you can identify, well, I keep seeing this chemotype that among my hits, so probably this type of compounds are more likely to be active than others. You still want them to resynthesize this, which is costly and long. And so what they do instead of doing this is something that's cheap and fast, which is to use their machine learning to select analogs that they believe are the most promising, that are commercially available.
Great, thanks for clarifying that, Matthieu.
Yep. And you can see that this method generated also some hits, a little bit more potent than the ones we saw on the previous slide, and probably because this requires an experimental, there's an experimental component to this approach, so it's quite different. But still, in all these results, again, I think it's very promising to see that we were able to identify hits for these underexplored proteins. In each case, it was a first-in-class hit for its target, and this was done within just after testing just a few hundred compounds. So that was that was very exciting to us. It is exciting.
It's kind of it's kind of neat. It's almost instead of relying on large drug target interaction databases or trying to, especially for low data targets where they don't exist, essentially Relay or Zebi AI is generating the training set that they need to build out their artificial intelligence or machine learning approach.
Yeah, exactly. Yeah.
So it's very interesting, but it does have that extra experimental step that needs to happen before they can engage with their engine. So very neat. Oh, sorry, I kind of jumped the gun there, but if you want to talk about the next big initiative.
Yeah. So yeah, because we are very encouraged by these results, we would like to expand this initiative and to make our experimental hub at HGC available to any computational chemist or AI expert in the world. And so to do this, we are starting this competition called CASP for critical assessment of computational hit finding experiments. And if we go on the next slide. So CASP, so it is a competition, and it's a benchmarking initiative to reveal the most efficient computational methods for hit finding and to guide future technological improvement and development. It is modeled after CASP, which is a protein structure prediction challenge, as well as a previous computational hit finding competition such as D3R. But on the next slide, you will see that CASP is quite different because it is a prospective hit finding competition. So we will not be looking at the binding poses of the compounds or the docking scores. We will be looking at experimental hit rates. We'll be looking at the potency, the diversity, the drug lightness of the hits, of the experimental hits, experimentally validated hits. So in this regard, CASP rather reflects a real world virtual screening drug discovery projects. So every 3 months, we will initiate a new challenge focused on a new protein target. Each exercise will focus on a new target that is representing one of 5 possible challenges, and which are defined by the type of data available, and I'll come to this in a moment. Of course, participants will be using their own computational method to predict up to 200 hits that will be ordered and tested experimentally by CASP at the HDC. And the first 2 competitions will be free to participants. We'll buy the compounds and we'll test them experimentally. We will cover the costs. This is an open science initiative, so all data will be publicly released without restriction on use. And of course, we expect that biotech companies will participate in this, not only prof, but also biotech companies will participate. And so we're not expecting that all algorithm will be open source. So of course, algorithm can remain proprietary. But we will ask to all participants to provide a sufficiently detailed description of the method that an expert in the field could learn from this and be inspired from this. So it can be a generic description, but not entering into very proprietary details, but still sufficiently explained. And participants will be anonymized, but the top performing groups will be de-anonymized at the end of when all the results are publicly released, as well as anybody who has to be de-anonymized.
Yeah, just very quickly touching on something you mentioned on the previous slide. CASP is, I guess, one of the standard protein folding prediction competitions out there, where there is a known, structure that's kind of blinded, and then a series of groups will predict that protein structure. And I think it might be kind of familiar to a lot of folks because of the AlphaFold performance of the CASP over the last few competitions, where that's really kind of been a shining beacon of how AI can maybe impact some of the studies we're doing. And so I'm hopeful that there'll be another maybe AlphaFold moment here, where some of the technologies that are emerging will show off to be very performant and really help accelerate not just hit finding, but also the initiatives of Target 2035 as well, right? So I'm eager to see what happens. And maybe a quick question about the participants. It's open to everyone who wants to participate, but is there a process for applying to the competition? Do you need to have I guess you mentioned the sufficiently explained methods, but is there more to it than that? Is there like a vetting process that people should be aware of?
Yeah, so it's all explained in detail on our website, and I'll provide the link later. But yes, so we'll have a multi-step vetting process. So the first thing, so we'll ask each participant to provide two example of papers published in the field, just to make sure that they have experience in the field. And then this is the first filter. Then the second filter is there will be a double-blind peer review where each participant will be asked to review 5 methods. So only the method will be presented, not the name of the participant, and not just the description of the method. And we'll ask other participant to say, is this method sufficiently described? And so for each method we'll have several participants who will have to say yes, no, it's sufficiently described or not. So that's the second process, and the third process will be an independent evaluation committee that will make the final call on the selection process. For the first challenge, we believe we have room for up to 25 participants.
Wonderful.
But we hope to grow. Yeah, we hope to grow in the future.
Thanks, Matthieu.
Yeah.
All right, sorry, I jumped in there. So a little bit, it's an exciting method, but please carry on.
So yeah, so today, if a scientist reads a paper on computational hit finding, or if a pharma hears a sales pitch from a biotech or disease foundation reviews a grant application on computational hit finding, it's very hard to compare these methods and evaluate which are heading in the right direction. And the reason for this is that all these people are applying their methods to answer different biological questions. So they're focusing on different proteins, using different experimental methods to validate their predictions, etc. So it's very hard to compare. So what we hope is that CASP will define the state of the art of computational modeling as it progresses over the years, and in doing so, will act as an accelerator in the field, really. Okay, so I'm going to be a little bit quick on the last few slides. I can see it's time's going quickly. So the target selection process will take into account a number of factors shown on the left here. Biological insight. So the target will be a target that is of potential therapeutic relevance, but that should be very innovative. So it won't be a target that or domain that is targeted in the pharma. In pharma companies, it's going to be different, novel targets. In some cases, we hope that disease foundation or pharma companies may be interested in championing a target and sponsoring a competition. Of course, targets we'll be working on targets for which there is, or we can develop cost-effective experimental assay, binding assay at CASP, and finally, each target will represent one of 5 possible challenges that are shown on the right here. So first challenge: a structure bound to a small molecule is available in the PDB, and there is some known SAR. The second type of challenge: structure in the PDB with compound but no SAR. Challenge 3. EPO structure in the PDB. Challenge 4, no structure in the PDB, but some known SAR. And challenge 5, no structure, no compound, nothing is known. So you can imagine that the expected outcome will vary depending on these challenges. So for challenge number 1, we'll be expecting some pretty potent compound, novel chemistry, and maybe a little bit of SAR. Well, in the case of the last challenge, for instance, we would be very happy to see any compound, even if it's weak. Yeah. If we go to the next slide, there's no question on this. So operationally, each challenge will last 20 months. So first, a target is selected, then virtual libraries are made available, and next, so the main panel in the middle. So the participants will have 2 months to run their predictive models, come up with 100 compounds that will then be procured within 3 months by CASP, and then CASP will take another 3 months to test the compounds experimentally and return the results back to each individual participant. And if compounds for a participant were experimentally validated, then this participant will be invited to select another 100 compounds that again will be procured at CASP, tested at CASP, and then will return the result back to this participant. And then all the data and description of all methods will be publicly released. So on the right-hand side of the right panel. And this 20 months exercise is repeated every 3 months. So there will be overlapping competitions. That's what you see on the next slide. Very, very briefly, schematically. Sorry, I forgot this. Sorry, the next slide. I forgot to mention that the hits will be evaluated based on traffic lights. I think, yeah, this one on a traffic light scoring scheme. So this traffic light will again depend on the type of challenge. For instance, this traffic light will account for the potency of the compound, their measured solubility, the drug likeness of these compounds, and the novelty.
I thought that last one was really interesting, Matthew, especially for the I guess the small molecules with SAR. I'm assuming that the novelty might be a little bit less, or diversity of compounds that you're seeing for those would probably be expected to be a bit lower, whereas the maybe hit finding, I'm not sure actually, but it's an interesting metric to be evaluating, I think. And is this done through pharmacophore, Tanimoto similarity, or there's it's an interesting traffic light.
Yes, we'll be using Tanimoto scores. Yes, yeah. But yeah, I don't know. I have no idea what we'll see either in terms of neurology, and I'm very curious.
Yeah, definitely.
And generative models will also be available, so people will be able to use either virtual screening or de novo design strategies. All right, so very briefly, the timeline for the first competition, December 1st. So actually, the call for the first call for participant was launched 2 weeks ago. So you can go to the website and apply already now if you want for the first challenge. And okay, I won't go through all of this, but basically, final results for this first competition will be released in October of 2023. And I can go to the next slide. Yeah, so and again, so each competition lasts 20 months. That's the big boxes that you see, but a new competition is initiated every 3 months. And on the next slide, yeah, this is the website if you want to know more. And on the next slide, so yeah, there is a white paper that is in press at Nature Reviews Chemistry, and that you can already access it through ChemArchive, the preprint. So you can see that many authors from pharma companies, Sanofi, Bayer, AstraZeneca, Novartis, Boehringer Ingelheim, Takeda. So you can see a lot of interest from pharma companies in seeing in which direction the field is moving, who are the main players in the field. But actually, the whole idea for CASP was originally from Alex Killish, who is heading computational drug discovery at Bayer, and the idea was really turned into a real thing, mostly thanks to Al Edwards, who's the director of the SGC. And again, the experimental hub will be supervised by Masoud Bedadi, who is also an author on this paper. And I think I have one last slide. Oh, yeah, well, I would like to acknowledge the hard work of my colleagues at the SGC and collaborators who worked on the AI pilot study on WD repeat containing protein, and I'm underlining the people who did the actual experiment at the bench. And on the right-hand side, I'm listing a few of the large consortia who are contributing to CASP. I'm going to stop here.
Fantastic. Thanks, Matthieu. We've had some really good feedback from attendees saying that it will be a promising platform and a great resource for the community. So thanks for sharing that with us. I think a lot of people on the call and people who watch it afterwards will find it quite valuable. We do have a question unrelated to CASP, if I may, because I know we have only 5 minutes left. So the question is, what sort of machine learning methods do you use for the screening? And this was in reference to slide 18, screening for target compounds that may have a therapeutic effect. So how does the model take into account the feedback about the molecule interaction with the target to fine tune the prediction? So that's quite it's quite a complicated question, so I can also write it out.
So this is a question for more for the participant than the organizer. I'm the organizer, and each participant will have their own response to this question. So maybe, I don't know if Vijay, maybe if you want to comment on this.
I could comment a little bit about the way we would probably use it within our machine learning engine. So with our particular approach, which is Matchmaker, we train it on a diverse range of drug target interaction data. So we train the model to first span the entire proteome. We're kind of the proteome first people. So we will take all the existing drug target interaction data that exists, map it onto protein structures across the proteome, and then from there, build out this generalizable engine that's learned from as many examples as it could of what molecule binds to what type of pocket. From there, on the proteins that we're interested in the case of WDR proteins, DCAF1, we pointed it to a few pockets that were assessed using, I think Matthieu mentioned sitemap. We used ICM MolSoft to do pocket finding, as well as P2Rank, which is an open source tool to identify regions on the protein first that were getting good druggability or DLID scores, if we were talking about ICM. And then we pointed the engine towards those pockets where it can then learn from the features that are there, predict what are the compatible molecules to those pockets, and those were the ones that we nominated and sent to Matthieu at the SGC to test. The question around target to disease link, that's a whole different field in itself. There's a lot of approaches that do it. There's some aggregate databases like Open Targets that's really great at linking the literature to targets between targets to diseases. And there's entire companies that focus on answering this question of linking disease to targets. And so don't think we have time to get into it here, but it's something that, not a specialty of Cyclica, but there are tools that exist that help you kind of prioritize and figure out what targets are relevant to what diseases.
Fantastic, thanks. I know that wasn't an easy question, so we're about 2 minutes left. There is, I think that's it from the audience. The rest were comments, but does anyone have any final remarks before we close up for the session?
I guess I would just want to convey my thanks to Matthieu for taking us through the slides today and educating us on the initiatives that are really exciting and in front of us, CASH, as well as Target 2035. And I think we're all very hopeful that, and I would say confident, if I could be so bold, that these are going to lead to some major impacts in the way that drug discovery is performed moving forward. So thanks very much to Matthieu for speaking with us today and sharing his insights. I learned a lot, and I hope everyone attending learned a lot, and looking forward to the outcomes from these great initiatives.
Yeah, and thank you to you, Vijay, for a nice discussion. I think it's and congrats to Cyclica for organizing this series. I think it's quite refreshing and novel. It's great to have this available online for viewing. Very nice.
That's great. Yeah, we'll be sure to post the link afterwards, and feel free to share it. And thanks again for your time, Matthieu, and to Vijay for taking the audience through such an engaging conversation.
Bye.
Have a nice afternoon,
Thank
everyone.
you very much.
Bye-bye.
Take care, everyone.
Bye.